Hello everyone, my name is Yinfei and I'm Head of Scientific Operations at Crown Bioscience UK and US sites. Crown Bioscience is a preclinical CRO providing high-quality models, technologies, and support for our sponsors' drug development needs. Today's 15-minute webinar is part of our educational Tech Talk series, focusing on in vivo solutions for oncology and immuno-oncology. There won't be a Q&A session at the end, but please feel free to ask questions in the chat box, and we will answer them as soon as possible. Today, I'm going to cover three topics. The first is the value of orthotopic and metastatic models, and then I'll follow up with how we set up oncology models using optical imaging. The last but not least, I'm going to quickly cover the advantages of high-frequency ultrasound for establishing oncology models. So, why orthotopic and metastatic models? These models can provide better understanding to mechanisms of disease. They help us to investigate the contribution of clinically relevant organ-specific TME to treatment efficacy, because these models will develop relevant tumor vasculature, stromal interactions. It has a right environmental pressure and also express common TME targets. They allow us to evaluate disease progression, something like tumor development, local invasion, and distant migration. It also gives us a chance to better understand metastasis, which is an advanced stage of most cancer and has a very high unmet need in cancer treatment development field. At Crown Bioscience, we divide our orthotopic and metastatic models into three main categories. The first one is orthotopic models. They are used to mimic primary lesion. They are established by inoculating cells into the organs they were derived from, say, liver cells back to liver, prostate cells back to prostate. This model will generate clinically relevant organ-specific tumor microenvironment, including vasculature, hypoxia, stromal infiltration, etc. They allow us to conduct experiments that will help us to better understand patient-relevant response, resistance to SOC and other type of therapies. The second category is spontaneous metastatic models. They are mainly used to mimic disease progression, including local regional extension, lymphatic and hematogenous metastasis, and also model progression from primary to late-stage disease. The last category is what we call experimental metastatic models. They are used to mimic end-stage disease. With them, we can model the adaptation of metastatic end-stage disease without restrictions associated with primary lesions. They allow us to evaluate the response and resistance to treatment in patient-relevant metastatic niche. So, optical imaging. So, at Crown Bioscience, we use the IVIS system, and we started with the very first version, the IVIS 100, and now we have 7 different equipment all the way from the early stages and the early models, Illumina 3 and the high-end spectrum CT. This equipment offers multi-layered research outcomes. They help us to progress drug development towards clinical trial a lot faster, and it has measurable economic benefits, include reduced animal costs and personnel costs. It stimulates insights into drug efficacy, kinetics, target, and mechanisms. It provides superior statistics, and the data generated are very reproducible. So this is the equipment we have. As mentioned earlier, we have Illumina 3, IVIS Spectrum platform, and IVIS Spectrum CT platform. We have used them for the past decade also and are very familiar with their characteristics, their capabilities, and what we can achieve with them. So in the next few slides, I'm going to use breast cancer as an example and to showcase a few models that has been set up using the IVIS system. Breast cancer is probably one of the cancer types that has seen the most advance in treatment. So a lot of patients can live a long time or even be cured. But there's still about 30% of patients with resected breast tumor will develop metastasis. So this is because undetected primary tumor cells disseminated into the human body. Although most surgical procedures are followed by systemic therapy, but these aren't always successful. Unfortunately, metastatic breast cancer are still incurable. Therefore, to develop new therapies, appropriate models that recapitulate various disease stages and show disease progression are essential. So there are three main stages of breast cancer. First is primary tumor, where the tumors are still confined within the breast, regardless how big they are. Then local invasion when they invade into regional lymph nodes. The late stage is distant metastasis, where cancer has gone beyond breast and regional lymph nodes. They likely settle in bone, brain, lung, and liver. So the scientists at Ground Bioscience have developed models that can cover each stage of this disease. To start with, and this is orthotopic breast cancer model. In this model, the cells were inoculated into the mammary fat pad. Data shown on this slide are generated using MDA-MB231, the triple-negative breast cancer cell line. So tumor growth can be followed with bioluminescence imaging. And so the 3 on the left is vehicle, 3 on the right is docetaxel treated group. This can be quantified. So as mammary fat tumors are relatively shallow, so we can measure them with caliper as well. So this is second graph. Of course, at termination, we can weigh the tumor. This is the one on the right. So this model will also develop spontaneous metastasis. So the instance of metastasis in lung, lymph nodes, liver, brain, and bone are shown in this pie chart. We can see there are very high rate of lung met, relatively high in lymph nodes, lower in liver, and relatively low in brain and bone. The MET lesion can be actually traced in life by shielding the primary tumor, but we tend to conduct what we call the ex vivo imaging, where we dissect the organs and conduct imaging outside the mouse body. Therefore, there's no impact from the primary tumor, which typically has a very high signal. This can be quantified, and the graph in the middle showcases the vehicle group has very high lung MET, and the docetaxel at 2.5 is relatively lower, and docetaxel at 5 mg/kg is much lower. Similar story as seen in the lymph metastasis. So this model, beyond primary tumor treatment, it can also be used to evaluate compounds that targeting, say, lung MET and lymph node MET. But if you want to study brain MET and bone MET, it becomes quite tricky because the incidence is quite low. To overcome this, what we have done is to inoculate the cells into the left ventricle of the heart. So intracardiac injection of MDA-MB231 will generate bone MET, and the cells was carried through the body by blood and they settle in the bone. And this can then be traced with BLI imaging. So ex vivo wise, we can do histology, X-ray, and MRI. So these two videos, the one on the left is from a mouse that basically had intracardiac inoculation of MDA-MB231. So multiple lesions will develop by about 3 to 4 weeks, and an endpoint typically driven by long bone lesion. This is characterized by BLI lesion intensity and clinical condition. The one on the right is treated with docetaxel, which effectively limits progression of long bone lesion, which no longer drive endpoint. But docetaxel has very limited CNS penetration, which results in the progression of CNS lesion, which typically drive endpoint, which at around 8 weeks. So if brain MET is the key indication that we're trying to test, we can also inoculate the cells directly into the brain. Data on this slide was generated using a human breast ductal carcinoma cell line BT474. The brain tumor was established by stereotactic intracranial implantation of cells. Then the tumor progression can be followed with BLI and also quantified with BLI as well. So this is just a quick flavor about what we can do with optical imaging. Obviously, there are quite a few more models to come, and which can be found on our website. So in the last few minutes, I'm going to quickly go through some data, and we have collected using the ultrasound. So last year, Crown Bioscience has acquired a high-frequency ultrasound, which is Vivo 3100 from Fujifilm. This system has extremely high resolution and can provide real-time, non-invasive imaging of mouse, or in that sense, rats as well. That's the largest animal we go. It doesn't require radiation or contrast agent. It can help with longitudinal studies. It measures anatomical and physiological parameters, extremely good at soft tissue imaging and molecular imaging. The data are very translatable to patients. A quick comparison. So the ultrasound we have has a frequency of up to 71 MHz. The typical ultrasound used in the clinics has a frequency of 15 MHz. High frequency lead to high resolution. Therefore, our ultrasound can reach a resolution of 30 micron, while the clinical one can only reach about 200 or 300 micron. In oncology field, they can be used in tumor detection and tumor measurement, and produce both 3D and 2D, angiogenesis, molecular imaging. One of the key application of the ultrasound is create orthotopic models using ultrasound-guided injection. So some very preliminary data actually from this one is from publication. So ultrasound can detect tumor as much smaller size. So on this slide, it showcases the data that tumors are detected from pre-palpable stage. And the mouse in this one is WAP-TAG mice. Those mice was generated by expression of TAG. So TAG overexpression is triggered by hormone stimulation, mostly during pregnancy. So following the treatment, most mice will develop tumors in about 8 to 9 months. But with ultrasound, this tumor can be detected much earlier. So if we want to do preventive treatment, this will be extremely useful model. And on this page is some publications, basically to showcase that ultrasound measurement can produce data at very high reproducibility, and also it matches MRI and CT. Another application of ultrasound is to detect metastatic lesion. Although we can do that using ultrasound, but that requires the cells to be labeled with BLI or bioluminescence. In IO field, that will introduce another layer of complexity, and the bioluminescence of fluorescence protein may have an impact on the immune system. While in this system using ultrasound, the liver met was detected in the red lobe of mouse, and we can detect met at about 2.7 millimeters and sub-millimeters as well. And so that doesn't even require any sort of label, and it makes life a lot easier. Okay, the last couple slides is another major application of ultrasound. So they can be used for ultrasound-guided orthotopic model setup. Generally, most orthotopic models are set up using surgical procedures, which takes about 20 minutes we have to open the mice, inject cells, and close the mice again, which is actually quite time consuming and also is extremely damaging to the mice. So some mice don't survive the procedure. With ultrasound, we can see in the video on the right, the cells are being, we can see the needle, the cells are being injected. This procedure doesn't require any anesthesia. There's no postoperative analgesia, no scar, and no inflammation response. And it also increases throughput. Then the tumors can be measured with ultrasound as well, and it's produce a very nice 3D measurement, and it's extremely accurate as well. So, but this data we have shown is actually using a BLI cell line. We just want to run a quick comparison between the surgical and the ultrasound. Both methods produce a very nice tumor growth, and they can be tracked with BLI and ultrasound, and the survival is extremely good, and the mice recover extremely fast using ultrasound injection. Okay, I think that's my last slide, and thank you very much for attending. This is just a quick flavor of what we can do with those two modalities. Please feel free to reach out if you have any questions. Thank you very much.